Started after my PhD, where I had worked on the regulation of enzymes, but the systems that I worked on were not terrifically interested, and I was looking for a more complicated regulatory system to work on. And so I went to work with Edmund Fisher at the University of Washington in Seattle, who had discovered, the first example of enzyme regulation by reversible phosphorylation. This was in the area of glycogen metabolism. In fact, in those days and for many years afterwards, phosphorylation was thought to be a specialized control mechanism confined to the regulation of this one process, glycogen metabolism. But subsequent work from my lab and many other labs, of course, showed that this wasn't the case, and that it was a very general control mechanism that regulates almost all aspects of cell life. And that led me to, to get involved in many aspects of this area. I found some of the first protein phosphatases and characterized them, some of the first calcium calmodulin dependent kinases. A big interest of mine for many years was how insulin regulates metabolism, and this was a 25-year project that finally came to fruition in the late 1990s when we managed to solve the signaling system. So this protein kinase cascade culminates in the inhibition of the kinase GSK3, leading to the activation of glycogen synthase, the enzyme that makes glycogen. For the last 7 or 8 years after that was solved, I decided to switch my area of research into something quite different, which I thought was still a black box, and so I've been now trying for the last few years to understand how signaling in the innate immune system works. When I switched my research about 6 years ago to the study of the innate immune system, it soon became clear that this process was regulated by an amazing interplay between phosphorylation and ubiquitylation. And these systems talk to each other in almost every conceivable way. So we had kinases that were phosphorylating and switching on E3 ubiquitin ligases. There were E3 ubiquitin ligases that ubiquitilated kinases. The formation of polyubiquitin chains caused those chains to bind to regulatory components of other kinases, which then switched those on, leading to downstream signaling. There were examples where phosphorylation provided the recognition site for an E3 ubiquitin ligase to then ubiquitilate and degrade another protein, and even deubiquitilases were themselves controlled by phosphorylation. So this is really how I became involved.